It's my great pleasure now to introduce you and Birney. Oh, but before I do, just a quick change to the agenda that Nikki Mulder could not be with us yesterday. As you heard, she's arrived and she will be speaking after Ewan. So we've shifted our agenda to deal with flights from Cape Town. And Nikki, welcome. So great pleasure to introduce you and Birney to do our keynote for day 2 of the summit. Ewan is based at the Genome Campus in Hingston, where over the last couple of decades, and probably a little bit more, has led a number of major scientific initiatives such as ENCODE, but also taken on increasing responsibility for the leadership of EMBL, EBI, and most recently with Rolf Apweiler being appointed the co-director of that incredibly important European asset for European Institute of Bioinformatics. Off campus, Ewan is a non-executive director of Genomics England, and most importantly from my perspective, being selfish, the chair of the Global Alliance for Genomics and Health. And for any of you who follows him on Twitter, you will also know he's one of the most prolific, eclectic, provocative contributors to the scientific social media. So great pleasure, Ewan Birney.
Gosh. So thank you very much. This is my first trip to Iceland. And I am going to work out how to drive this. How do I drive this? Perfect. And I asked actually Eric Green whether I should cut my cheesy slides at the start. And he said, no, don't cut the cheesy slides. So the talk is really in 2 bits. One is a little piece of scene setting, and it's no surprise, but I do think it's, I don't know, to channel Eric, it's worth stepping back out why the world is changing. And then we can step back into what is quite a technical area, frankly, which is how to set up standards, technical standards for genomics. So we've been living through a remarkable revolution over the last 20 years. This is my preferred way to describe that revolution. In 1999, an American raised enough money from the US stock market to sequence the human genome. We later discovered it was his own genome, and it cost the same amount of money as the most expensive house in London that was sold that year. Now, this is not a picture of that house. I'm not allowed to take pictures of that house. This is a nearby house in Kensington there. These days, if you wanted to get your genome sequenced, it costs substantially less than an Arsenal season ticket. It's unclear whether this season it is more valuable to have an Arsenal season ticket or your genome sequence. I think we'll discover over the next couple of weeks whether this is true. Depending, I actually don't know the costings of the Icelandic football team. But going to a different location, it costs about the same amount of money, again, as a Yankees season ticket. Many people have an Arsenal season ticket. Many people have a Yankees season ticket. They could have had their genome sequenced rather than participate in that sporting aspect. And this is an example of how genomics have become a billionaire's process to frankly something that you can imagine everybody having. Now it's had a big change to the way we do basic research, and I have a whole talk about this which I will not give you, but just to say basic research has moved from mainly being advanced cookery, which is the stuff that you do inside of the wet lab, to at least being half and half between how you manipulate things in the wet lab and how you draw inferences from it in computers. So biology has become an information science. You have to lace your research process with computer scientists, with information scientists, data scientists to draw inferences. There's a whole story about that. But it's also having a big impact in this place between research and medicine. And now many of you know this, but again, it's worth stepping back. The process of research is very different from the process of medicine. And actually, if you're from an English-speaking country, you can miss this easily because of this very first fact. For English-speaking countries, we are lucky that the international language of science, which is English, happens to be the same language as the language we practice medicine in. And so we do not see this transition in such stark detail. If you are not in an English-speaking country, you will know this because your patients will not speak English just to make sure that they can communicate with the scientists around. They'll be speaking whatever your native language is. The cardiologist in Berlin must speak German to his patients. The cardiologist in Berlin must have a very, very good working knowledge of German, if not be German. So this national language aspect is actually for me just a marker. It's a kind of indicator variable. When I go around to my colleagues in Europe and I talk to the medical side, in fact, all the conversations happen in English. It's very lucky that my native tongue is the international language of science. But you always notice it because the directions to the room is in German or in Spanish or in French. And so I get lost and I have to ask people where I'm going. And it's a very clear moment when you transition from the science world into the medicine world. But actually, the more important things are below that language aspect, the amount of legality, very different systems across healthcare around the world. We organize healthcare in a very different way, much more focused on closed data. And the final thing, so grants and contracts. If you're a researcher, you have your first paragraph, you say, I'm going to research this and I'm going to cure cancer. The funding agency does not come and sue you 5 years later because you did not hold up that first paragraph where you said, I'm planning to make a substantial impact on cancer. If you take a contract from a hospital to process 10,000 blood samples and you do not process 10,000 blood samples, they will come and sue you for not processing 10,000 blood samples. So the mentality in these systems is very, very different. Here are two systems. One is the left-hand one, which I'm most familiar with, which is the one in the UK. I will not attempt to pronounce that incredibly long German word. But just to say that the kind of fundamental wirings of how healthcare is done in different countries is very, very different. Human biology is incredibly universal. Human healthcare is very diverse. And in this world, between a world of research and a world of medicine, it became clear to us, in fact, a decade ago, that we have to build bridges. And actually, just selfishly, from my position at the European Bioinformatics Institute, we are research-focused, international, English as our working language organization. We are just not the appropriate organization to go all the way into healthcare. And we realized that we had to build partners that we could transfer our technology to across, in our case, Europe. And there is now, it's just, I think, something to almost to celebrate, which is how deep the engagement of genomics into healthcare is. So I'm a non-executive director of Genomics England, and so I know that one particularly well. This year, the NHS England have said that rare genome sequence can be a routine part of medical practice for certain conditions, in particular rare disease. And so there's now a process for genome sequence to be routinely asked for by doctors, processed, and put back in to the healthcare system. My colleagues in France have an ambitious plan, same sort of scale as Genomics England, and they have selected the first sites to deliver that work, Plan France Génomique. We're in Iceland. So by the way, there's a interesting perspective here. The smaller the country, the faster the prospective research cohort and your medical genomics merges. So the Estonians are probably the best example of that, in which there is very little distinction between the medical genomics effort and the population cohort effort. It is one effort. The smaller the country, the more these two circles overlap. The bigger the country, the more these two circles stay separated. At some point, they will overlap completely. We built, so a final thing I should mention about healthcare is it has a very different scaling property than research. Healthcare is an expensive process, but it's also a process that has an awful lot of money. And importantly, it is much more about whether a particular procedure is justified. Is it justified from medical grounds? Is it justified on health economics grounds? Is it justified through the regulatory process of that country? Once that is justified, it normally gets rolled out pretty reasonably consistently across everybody who fits that criteria. And this means that healthcare will scale to a far, far, far bigger number of humans than any research process. It ultimately scales to every human on the planet. And we built a simple, rather crude model of how we think this is going to work out, in fact, about 1.5 years ago. The orange line at the bottom is the number of genomes we think will be sequenced around the world due to rare disease genetics. And the blue line is on cancer. I'm actually much more confident about the orange line. The orange line is now really a question of implementation across the world. The blue line still has a lot of what you might call clinical research about what precise mixture of genomics assays are the best assays for cancer. And for the people who work in that space, you'll know that it is actually quite a complicated question. It's not so easy. For rare disease genomics, it's pretty clear-cut. The important thing to look at here is the scale on the left-hand side. The scale is in tens, hundreds of millions of people. And that is just not that's exactly what you expect from a process, healthcare, which runs in all the countries of the world and wants to serve all the people in those countries. So this is a process that will deliver incredibly high numbers of sequenced human genomes. Different way of doing this, we made these estimates, sorry, it should say 2025 on the right-hand side, of the percentage of whole genomes and exomes that are funded by healthcare systems. So the moment we think about 20% of the world's capacity is going towards medicine rather than research. And we think by 2025 it will flip around so that only 20% of the world's genome sequencing capacity is going to research. Now, for researchers, I would like to introduce you to this remarkable organism, this bipedal ape. They are great. They're a very standard mammal. If you want to understand mice, they're an incredibly good model for mice. But importantly, humans not only are great models for human diseases, but we are a very standard mammal, in fact, a very standard metazoan, and we have all the components of life that one wishes to study. And we are an exciting resource to use via this process of volunteers in different ways. So we're outbred organisms with pretty good genetics. We have a slightly irritatingly rapid expansion across the world, and that means our LD structure is frankly a little bit too long than you would like. I much prefer my Japanese rice paddy fish or my Drosophila, much tighter LD, much, much cleaner genetics. But you can get insane numbers of humans, and they walk into these phenotyping centers called hospitals, and they self-fund their phenotyping through this process called healthcare. And so they're a remarkable, remarkable resource for research, not just for research into human disease, but also a resource into basic biology. And if you look at what studies like the UK Biobank are doing in this space, it is remarkable. I think as much of the research will be on basic fundamental properties of mammals and physiology as it will be about human disease. And that's to be celebrated in many ways. So I hope I've convinced you that we also have this incredible opportunity of not just these research cohorts, which you represent, but also this broader process of healthcare taking us to tens of millions. So we need to be able to responsibly share data globally. And that's how I got involved in GA4GH, the Global Alliance for Genomics and Health. So our overall mission is to accelerate the progress of human health by establishing a common framework of harmonized approaches to effective and responsible sharing of genomic and clinical data. I think our best analogy for the more geeky people here is the W3C consortium. So W3C consortium, it's a standards body that creates the standards for the web, HTML, HTTP, HTTPS. If you don't know the W3C consortium, don't worry, but you use their standards every single day. When you woke up and you switch on your mobile phone and you looked at a website or you checked Twitter or you did any of this, you use the W3C standards then. GA4GH is aiming to be the W3C-like organization for genomics. So one moment here, there's a little bit of language shift. We use genomics in the broad sense, so all these genome-wide assays, genome, transcriptome, proteome. We're in the midst of this transition from a research commission genomics data to healthcare commission genomics data, and I put down that we have 2.5 headaches. So the first one is the healthcare community is not used to this data, and there are some challenging aspects to it. It's quite technical as well. Healthcare data is not routinely and broadly shared, and it's unlikely to be shared at a population scale. It's unlikely the case that all of the Danish genomic data can be slurped up and put on some very big disk and shipped to Shenzhen or Washington, DC for the pleasure of some researchers to fool around with. There's quite a strong need to respect the jurisdiction and the regulation of these incredibly large data sets. And the final point, which I call half a problem, which is that we have hit some internal scaling problems inside of genomics, and we actually need to retool. It's a kind of natural part of our process that some of the methods that worked fine 5 years ago or 10 years ago no longer work, and we need to go through a process of improving that. So I just want you to step back a little bit and ask the question, do we need this? So it's interesting to ask, in particular for the large countries, US, China, the EU, if you like to call it a large country. Could you just do it inside your country? Could you just coordinate nationally and not have to coordinate internationally? And even for the biggest countries, this will not work. The allele frequencies of interesting variants around the world is too much. There's actually an interesting other problem, which humans have always migrated, mixed, and had children, had sex. And therefore, even in these large countries, the population that's inside of your large country is not necessarily well represented somehow just by randomly sampling from your country. You need to understand the diversity in the world even to treat and understand the people in your country. We have a concrete proof in rare disease. For the people who work in rare disease, this is bread and butter. They work internationally because very often there are only 4 people with this particular mutation around the world, and they need to find those 4 different children, phenotype them and genotype them, and to make a diagnosis. So in the rare disease world, this is already accepted. You cannot do this in a European or US or China only way. You need to work together. But I think that this is going to be increasingly true beyond just rare disease. Another aspect of this is how we handle the ethics and the legalities of this. And it's really important to realize that there's going to be a shift in behavior. Researchers are not going to get access to this data in the same way that they download data sets at the moment. So jurisdiction is tightly coupled to physical location, and one just has to accept that as a constraint for this. And then again, this is this 2.5 headaches, which is we've really got to get away from file access. It's killing us in our pipelines, and we have to go into a more modern world where things are services, not files. So the solution is to federate, so move analysis to the data, not aggregate data close to this, have broad reciprocal data access. So basically, by trying to have an international community that does this research, we're trying to say that researchers can get access to these different data sets, but those data sets will stay under the jurisdiction of each country. Transfer skills to the healthcare setting and make more discoveries. So just to emphasize this, at the moment, open research data is aggregated to single points around the world. EMBL EBI is the European point of this. And then you download that data set locally to do that analysis. And for the people working on plants or on mice or on diatoms or on other things, this modality will, I think, continue and is the right way to work. But for humans data, this is not going to be the future. The future has to be that one takes your analysis and one sends those analysis as containers into different data silos across the world. So this is my fantasy world. Here's a researcher at the Sanger Institute just over the road from EMBL EBI. And she has developed a whizzy new analysis method. She gets very excited by the results, but she realized that she needs 100 million ascertained genomes because it's prostate cancer and smokers, but they have some other rare germline variant or something like that. And so she needs to have a much bigger data set. And at this time in 2022, Sanger only has 1 million genomes that it can access directly on its data sets. So she now realizes Sanger has been compliant to GA4GH standards, so she knows that her code can run. And she now spends, so in a fantasy world, this will take just a click, but I'm a bit more of a realist. So I'm going to say it's going to take her 2 weeks of porting for each of these 4 data islands in America, at all of us, the UK, France, and Denmark, so that she can go and run her analysis across all of these clouds. Each of those analysis produces a sort of intermediate results file, which are allowed by those clouds to be taken off. And she combines them, publishes a Nature paper, and cures cancer, which is brilliant of her. This next example actually runs now, which is about rare disease genomics. I'm giving an example here of a pediatrician with a rare disease assay, and she goes and looks through this matchmaker exchange and gets a match, and then from that match, matching 2 individuals can confirm the diagnosis. The parents get cancelled, and the gene enters this association. This protocol, which is called Matchmaker Exchange, is a bit clunky, but works now. Now, Peter, how much more time do I have? Not much. Oh, brilliant. Okay. The lights changed, and 8. Okay. So that is the vision for GA4GH. How are we actually internally constructed? Well, you saw some of the slides from Thomas Keane yesterday of our driver projects and our work streams. Driver projects are listed here with the different logos, and the countries that the driver projects touch are placed in blue, which is really nice. You can see there's a good spread of countries, and in particular, this year we made a big push to move outside of the Anglo-speaking world. And so I'm very proud of the fact that we have the Swiss Personalized Health Network as a driver project, the H3 Africa project, which Nikki, I'm sure, will talk about, and the AMED project, umbrella project in Japan, are all now driver projects to GA4GH. So we properly have a global spread. You do not have to be a member of one of these projects to participate in GA4GH. This is in no way a requirement for participation. It's just to make sure that we, the people who are making these genomic standards, are grounded in the realities of delivering this for these people. So it's a way of grounding the technical experts across GA4GH. And I now have a series of, there's no way you can make technical standards look sexy and cool. These are some of the technical standards that have recently come out. be reassured that if you're someone who produces these or uses this, you're like, oh yes, that solves a problem for me. So this is a rather straightforward standard, which actually frees us up from file formats and allows one to get access to data sets as a service rather than to get access as data sets by asking for a file access. Now, you might think that that's an incredibly small change. It's actually the smallest change needed to enable a future world where we can do cleverer things in that access process. This is such an interesting standard. So this is about knowing which reference human genome you are using. You might think this is an easy problem. It is a surprisingly hard problem to ensure this. You might think that just these version numbers of the human genome could do it. You'd be wrong. I'll introduce you to the Y chromosome PAR region, which gets processed in all sorts of different ways by different people. And so even understanding, have I matched to the same reference genome as you have, is a complicated thing. This standard and API handles this with the modern computer science way of hashes, which basically work extremely well. It's the way most of the internet runs now, is by exchanging hashes. Here's another standard. This is around the ability to computationally indicate what consents have been signed by the patients and what usage researchers want to make of a data set. So using this computational framework, you can express the patients consented for research on diabetes, and both commercial and non-commercial researchers can be used. And so this is now in use in a variety of locations. There may, at the moment, the goal is to make the process, which is currently done by literally writing this longhand, having someone read it, think about it, go to a committee and turn up, is trying to make that process smoother. It's not impossible to imagine for low-risk scenarios for a completely automated flow. For high-risk scenarios, I doubt we'll ever take a human out of the loop in granting access to people. But for low-risk examples, I would like to get access to the height data set from the South Korean population. It may well be that the South Korean ethics committee is like, we are happy for that request to be executed automatically through this. Many of you will use this. This is a file format, but it represents two different things. It represents an incredibly efficient way to store data. So this is the CRAM format for raw DNA sequence data, and it's also updating itself for some of the newer sequencing technologies to be able to represent very long reads or DNA modifications in the future. So this is right at the guts of everything we do. If you're a bioinformatician, it's probably the bane of your life handling these things. But here we have improved the way we store data at the physical byte level. And this is a discovery process. This is an interesting project. The Beacon API was set up almost as a demonstrator project to say that people could share. It was considered the simplest thing to be able to ask another data set, which is, do you have this allele in your data set? And the answer is yes or no. What it's become is a variable discovery stack. And the interesting thing they've put in there is this idea of registered users. This is through authentication and authorization processes who can ask richer questions than these very simple questions on these data sets. And again, something to think about in the future is I think we're going to have a very risk-based approach in some of this, which is exactly the way the financial industry thinks about this, exactly the way computer security people think about this, where we vary our level of paranoia relative to the risk of harm for the different different access routes. I will not bore you with this. It's yet another, well, maybe I make one comment about the VCF file format. Probably many of you, again, or your colleagues use this. This is one of these things which is really not scaling at the moment. This scales very, very badly. And we are going through a process of completely gutting in this, or at least evolving this in the future to make this scale correctly, because there's no way that this system will scale to millions of people. GA4GH sits in a kind of ecosystem of standards. You'll realize that we're not the only standards organization on the planet. At one level, we're interfacing with healthcare, and there we have a very good collaboration with the HL7 group, which is the group setting standards for electronic healthcare records. They find genomics data confusing and annoying. We find healthcare data confusing and annoying. So we have a common cause. And so we have made a common work stream where we are informing them about how to represent genomic data in healthcare records, and they are informing us about healthcare data. There's a kind of granddaddy of standards organization called ISO. We're part of one of their slightly complicated cross-ISO groups. And where appropriate, we'll take our standards through an ISO process, which for some people makes people happier when you can say it's ISO standards. And then there's a bunch of technical standards, which I'm not really going to go into, but there's things like authentication and authorization. There, we are not reinventing anything. We are hooking in to the correct, well-established industry standards for authentication and authorization and adding the necessary things we need to make those work for us. For example, this data use ontology would be an excellent example of something that we need to own and handle, but then it gets attached into relatively standard authentication and authorization. So kind of my final slide, almost final slide, just a comment about IHCC and GA4GH. In some sense, this is an incredibly well set up by design process, not least for the fact that Peter is involved in both of these things. So I don't, all of these things you could see from the presentations of Thomas and of Anthony yesterday, that all of these processes are aligned. This just gives you, I hope, a broader picture of how GA4GH thinks about the world. Large-scale cohorts is a strong community for the standards. You should be big pushers and drivers for them. You should be big users of them. You are at the moment. You should continue to be so. And excellent links to what I didn't realize how you name yourselves, but Team A sounds great. Go Team A. You can talk to me about GA4GH, but of course you can also talk to Peter, who's the chief executive of GA4GH. Peter's team runs an incredibly good shop for delivering all of this. And just to mention that there is an external plenary open to all, and it's in the US, in fact in Boston. It's not near ASHG. So it's near ASH, it's near time-wise and vaguely near geographically in a Houston-Boston kind of way for this. You do not need to be a member of the Driver Project. You do not need to turn up at these plenaries to get involved. We are very much an internet-run organization. It's completely open. You go to the website, you go to Zoom calls, you get on mailing lists, and you participate if you want to be involved in the actual technical aspect. So thank you very much for listening.
Questions
Eric?
for you, Ed?
Eric?
Well, first thing, great job listening to the person who gave you advice for the first half of your talk. No, but it did help. I really think it was very, very well said to set a context. My question is, I really like the analogy that you folks make to the world, from what I understand about the World Wide Web Consortium, and how the parallels I think are very striking. Have you? I don't know the history of the World Wide and how it evolved as an organization over time as it accomplished its goals, but then maybe needed to do other things when it reached a more mature state? And have you tried to think
We-
about the evolution and maturation of your organization in parallel?
We've spent, it's one of the organizations that we've looked at to see how they work. There is a slightly different dynamic because commercial organizations are such a big part of that world. So whereas our world is healthcare organizations, and healthcare organizations are a slightly different beast from commerce. So I think that's the only place where you can't just draw a one-to-one mapping. Yeah, but there have been a big source of inspiration. Our current operating model is very similar to the W3C consortium.
Yeah, Mike Asiano, Million Veteran Program. I think that was a brilliant laying out of a number of bridges, and I think good population scientists, I completely agree, need to have systems biologists, basic scientists at the table to design good studies. I want you to comment on just one other area of standardization that you alluded to. We have a Victorian structure of disease that's based on organ systems, and I really feel we need the same kind of investment and enthusiasm in mapping building a better map of the human phenome. It's wonderful work on the standardization on the omic side of the equation that we need, but I think we need that same kind of investment.
Yeah, I mean, I couldn't agree more. And I don't know if you know my colleague Helen Parkinson, who heads up this area in the EBI, who takes an incredibly, I think, practical but also quite visionary approach to how to handle this. It is mixed up in a lot of things somewhere between, this ends up being very highly granular ontologies versus, a kind of deep learning, quantitative transfer learning problem. You can talk about these as latent spaces and a transfer learning problem. And I think we need both research and then a process of taking that research into practice for this. And I'm just very aware that we are kind of one part of that process, if you'd like to put it that way. There has to be a very clinical research component, and then there has to be clinical practice as well. So all three of those need to be together.
Right, and there's a lot more history.
Yeah, there's a lot of history there. Absolutely.
ClinPheno working group there.
Sorry?
The ClinPheno?
Yeah, ClinPheno. We have a work stream precisely set up for this space between these different areas, which is called the ClinPheno working group. But again, that's actually standardization. And I think you're talking more about the research side, where you kind of change the paradigm for a particular disease. And that for me is seated inside of clinical research. Anthony?
Anthony, Philippakis Broad Institute. I really liked your analogy in the beginning about medicine and research and the difference between the two and the need to build bridges. There is one activity that truly is care delivery and research at the same time, and that's the world of clinical trials.
Yes, ones. Absolutely.
And because we truly are engaged in a research activity, but we are delivering care, treating disease.
Yep.
Why do you feel that genomics is not more a part of the clinical trial apparatus? Because you would imagine that in a large-scale clinical trial, you may spend $20,000 per year per patient. To spend an extra $100 to exome sequence doesn't seem so crazy. And do you feel that should be maybe more where we put our attention?
So there's two things. One is, from a standards organization, we have had good relationships with CDISC, which is the kind of area there. Actually, I think we can go back and try and recreate. Oddly enough, we've got a much more established thing with HL7 than CDISC. Your question is more a kind of trials question, and I'm slightly sad that Rory isn't here to answer that, because I think a trialist would say, Rory is there, so maybe Rory can answer that. I think a trialist will often say, you have to understand your randomization and what your data is going to be used for, and basically fishing expedition, the whole bloody point of clinical trials is not to do fishing expeditions. Fishing expeditions happen somewhere else. And when you're in a trial setting, if your trial protocol needs genomics, so be it. But if your trial setting doesn't need genomics, you perhaps shouldn't add it on. I'm channeling you, Rory. Why don't clinical trials use more genomics?
Well, I think a lot of clinical trials now, actually over the last 10 years, have stored sample, and increasingly they're being used and misused. So there have been a lot of really lovely examples of subgroup analyses based on genetic data in clinical trials that have failed to understand the problem that you just highlighted of fishing expeditions. I would wager that the genome risk score predicting the relative risk reduction in cardiovascular events with statin therapy is one such fishing expedition that got the wrong answer, in the same way that, say, KIF6 did for statin. It gets rolled out into clinical practice based on subgroup analyses, which are no more reliable than astrology.
Yeah, okay. It's a bit harsh. But I think the meta problem here is that you've got to separate out your test from your hypothesis generation needs to be a separate process from your hypothesis testing in clinical trials, and you have to keep yourself squeaky clean about those two things.
Thank you very much.